All right, thank you so much. Why don't we have all of our speakers come back up and line up over there with Tori? That'll make it easy for questioning. And then if anyone has questions, please head to the mic. Do we have it online?
There is one question for Tori. Tori, what type of device was used for coring? A deep core will take a lot longer than the times you show.
Yeah, so the deep core was done at the LSEP, and those values we didn't consider times at the LSEP. This was just the drive core. So it was the smaller model, and that was the one that they brought to stations and then did at the station.
Let's go over here.
Hi, Aisha Katib from UMD. This is also for Tori, sorry. So I was wondering about how you managed to define the start and stop times for all of these different astronaut activities and what your margins of error were. Because and also as a follow-up, what would you suggest for Artemis 3 astronauts or Artemis III to kind of track these different activities in the future?
Yeah, so in terms of what we use, so basically over the past few years at LROC, we've been working on spatiotemporal maps, which are basically like, okay, we know that the astronaut will appear at this time, and it's mostly based on the transcripts. It's also based on the video, pictures, that sort of thing. So a lot of that came from our own data set, but we use like the ALSJ as a resource. We used Apollo in real time as a resource. So these were all, it was all like stuff that we collected. If we didn't have the data when I was compiling all these times, then I was looking back at those other resources. I think that answers the first part of the question. Yeah. Okay. Sorry, can you remind me of the second part?
So if you want to do this analysis again with Artemis, after we track Artemis astronaut activities with Artemis, what would you suggest to kind of make the process easier and build on what Apollo's already kind of given?
Yeah, having lots of cameras everywhere is like the big one. I mean, I think ability to track, whether like GPS, whatever, that would be really helpful. Just a lot, like it was really great to have the video and the photos, but just increasing that, having video inside the astronauts' helmets would be great. Like, just it's possible to piece these things together, but having a lot more like video is really helpful. Basically, is what I would say.
Thank you.
Yeah.
All right, thanks, Carl Hibbs, APL. I have a couple of questions, but it'll be for the radar talk. This one will spread the will. So you showed a lot of slides. Do it quickly, but one thing I keyed on was, believe it or not, the backscattering effect. It looked like you showed a 2-degree wide backscattering effect. And you mentioned that can give us some insight into the volatile content in water.
Right, so
Can you explain that a little bit more?
About extracting information about volatile content?
Yes. How one can use the backscattering radar effect in this.
Yeah, sure. So
Thank you.
there's multiple ways of doing it. One is to sort of deconvolve the sort of permittivity and dielectric value for the subsurface, and so therefore you're assuming a certain percentage of ice within the region you're viewing, and then you're comparing that permittivity from value from what you'd expect from say a basic dry regolith. It's kind of tricky on the moon, that way of tackling it, because lunar regolith is around 3 or so, and sort of solid ice would be at a dielectric of like 3.15. But there's ways to tease it that way, and a DSR team are using these sort of inversion models from the backscatter to try and assess that. That's one way. Another way we're doing it from the bistatic campaign as well, is into this opposition effect. And basically the idea is we take a measurement in the same area, looking over larger and larger bistatic angles. And the idea is you should have this sort of uptick at low bistatic angles, which could be indicative of the presence of ice. There's been modeling work going on with a colleague, Parvati Prem, to sort of look at that, and also analogs using optical wavelengths have shown similar sort of ideas. So those are 2 ways we're attacking it.
Thank you.
No worries.
Hi, my name is Kristen Lusinger, NASA Ames. My question is for Heather. And my question is, when you're looking at survivability, what sort of timescales are you looking at? Are you looking for like indefinite survivability? And one of the reasons I ask is because I like to say we're very interested in the moon over geologic timescales. Missions operate over human life scales, right? So survivability of a microbe over the course of a mission might be different than survivability indefinitely. So what would happen to those maps if you potentially look at seasonal survivability or, shorter timescales?
Yeah, I think that's a great question. And I think the first thing I would want to say to point out is a lot of you speak of geologic time. We don't have really great data for spore survival for many of these taxa. Usually people give up after a decade and they're like, I guess it's dead, and so we don't actually have a lot of that data, and that's probably not something we could do. And, that would be hard to get. But yes, many of these cells, even if they're not viable, remain around in the physical environment. You can pull them out of a, liquid nitrogen dewar a decade from now, and they'll be sitting there just fine, ready to contaminate your light element analysis. So I think that's the first part of what your question was, and I honestly forgot what the second part of your question was already.
Oh no, that was perfect. Thank you.
Okay, cool. Yeah.
Can we check in online? Do we have any questions online? Okay, go ahead. Let's go to this way.
Hi, thank you so much. I'm Artash, a student from Toronto. I definitely learned a lot in this session about the potential for different areas to explore in the Artemis III and future missions. But I had a question a bit from the perspective of the next generation, as I'm in my last year of high school. So a lot of these areas clearly have a lot of potential for research. As Heather mentioned in her presentation, potential for microbial life, especially the South Pole region. So just like back 50, 60 years ago, when we had the lunar missions, some of these samples, or actually the majority of the samples collected on the moon, were preserved and locked away for future generations to analyze. So similarly, I was curious to hear your take on whether some of these, high potential zones should be kept unexplored and uncontaminated for future generations with more advanced technology to explore.
All right. Do any of our panelists want to tackle that question? Does Jake want to tackle that question?
I think that's a really sticky question. I think that one thing that I would say before I turn it over to Jake is that there is a custom in other fields like archaeology of leaving something of every site that you explore, a portion of it always unexplored. You only do a partial dig at any given site so that you do leave something for the next generation and their technology. And I don't know if we really have that in geology or in planetary science, but that is a practice in many other fields.
Yeah, this is a really great question. And I think this is a really good time to be asking this kind of a question because we're on the cusp of just beginning to explore a place that we may be going back to multiple times. Hear about some of the reasons that these places are unique and maybe delicate in some ways. So these questions are definitely being asked. As I mentioned this morning in my presentation, this starts to get into concepts about regulatory aspects. How do we start to make agreements with partners about, what is the access to these areas and how much will we be doing in these areas as we move forward? So, I don't know that we can say a definitive answer to your question, but I do think this is a topic that we'll see being discussed more and more as we move forward. And the more that we learn about these sites from multidisciplinary approaches, the more we'll understand processes, are things that need to be protected in the South Polar region, globally, between both poles. I just think this will be a topic that we'll have to discuss moving forward a good bit. So yeah, thanks for that really good question.
Thank you.
All right, let's head over here.
All right, this came off. Hi, my name is Nandita. I'm a grad student at Stony Brook University. This is kind of a question for Julie and Jenna. so more of something I'm really curious about. So you guys presented boulder maps of separate sites. So I am curious, how does that compare to amount of boulders you see at other Artemis sites? I'm saying this because a team of people I worked with in 2020, we published like a detailed map characterizing boulders at site 007 and 011. And back then we didn't have like a site to compare it to in the nearby region. So we compared to north and south rim craters from Apollo to like look at the distribution. So I was wondering if you have done something similar. Oh. Damn it. Is this better? No? Should I go to the... Can you hear me? Oh, this is better. So my name is Nanta. I'm a grad student at Stony Brook. I think you guys presented boulder distribution at 2 different sites. And I was wondering if you have done a comparison in the distribution amongst the sites, potential sites of Artemis 3. I'm asking this question because a team of interns that I worked with in 2020, we published an article last year, kind of detailing the distribution of boulder sites at site 007 and 011, which are the selected sites among the final sites. So back then, when we did the detailed mapping, we didn't have anything to compare to within the proposed Artemis sites. So we did a comparison with North and South Ray Crater because they were Apollo sites. So I was wondering if you guys have done something similar in terms of numbers, and what would you expect? Like,
okay, I think we got the gist of it. So we didn't really do a full up like boulder count. So we identified boulders that we could identify in the images for the one site. Jenna went through and identified a lot of craters, in particular areas that we thought would have interest for specific volatile reactions. In terms of whether that has been done for all of the sites, I don't honestly know. I think people are still working on it, at least within NASA and maybe other folks in the community as well. So I think like this sort of meeting is a chance for us to kind of all come together and find out what people are working on and where we can collaborate. So if that's something that people are interested in, we can help. But yeah, we haven't done anything like that ourselves.
thanks. Hi, Alana Lee from NASA Goddard, and my question is for Heather. So in some of the taxa that you were describing, have you considered impact resilience, and if that could be used as an analog for potential landing sites and disruption that could be caused to either existing taxa or introduced taxa in terms of mitigation strategies?
Sorry, can I get you to repeat that? I lost what you were saying right after the word taxa. It's super echoey up here. I'm so sorry.
Is it better from here? Is that better or worse?
Yeah, maybe.
Okay. So I'm asking if you're aware of whether they're resilient to impact events. So obviously in nature, that would be cratering events. And I'm asking, can that be extended in terms of when we're considering what types of disruption could be caused by actual landing events? Do we know if there's resilience in terms of survival? And how can we think about that in terms of mitigation for either existing taxa or introduced taxa?
Yeah, so I think you're asking about the survivability of things that might be transferred through an impact event. Is that correct?
Yeah, or even just survivability from an impact event.
Yeah. So there's actually a group that's done work on this to try and understand how survivable it is. And their idea was thinking about the idea that, you know, there may have been microbes swatched back and forth from Earth to the moon over the course of our histories together. This is a group in the UK Space Agency. They actually have this rail gun or something that they use to shoot microbes at things. And most of them survive just fine. It's usually the kind of organisms that are spore forming, although not exclusively. There are non-spore forming organisms that can survive impacts, just as there are non-spore forming organisms that can survive open space. So they have really novel mechanisms of doing that we're just now starting to really understand. Some of this has to do just with the structure of the cell, and some of this has to do with abilities to regenerate really quickly, which also helps them survive the radiation. So there's a couple different things going on there, but yeah, impacts. Some things do just fine.
Great. Thank you very much.
I hope I answered it.
All right, these are going to be our last two questions. We'll start over here.
Okay, thank you. Brian Day, NASA Survey, also for Heather. You make a compelling case for looking at polar regions, but have you given consideration to taking a look at lunar module impact sites? Because they did carry biological waste material that we would know precisely when it impacted, and we'd be able to measure degradation and perhaps even survivability.
I am incredibly interested in looking for microbes at just those kind of sites because, yes, depending on, since some of these light and temperature things can remain very static for a very long time, those could be perfectly survivable places. I didn't mean to intimate that only the poles with their low angle and their surface roughness would be survivable. That's just where we focus this study on, since this is the current area of exploration and especially human exploration. So yeah, you could take those same sort of relationships of radiation, temperature extremes, water availability, volatile availability, and do the same sort of mapping activity over very small areas to try and understand where there might be a survivable niche for a microbe.
Thank you.
Hi, Parvati Prem, APL. Thanks everyone for really great talks. Really enjoyed the session. I have a question for Jack. So you had that map where you showed some M-cubed water detections coinciding with that peak ring.
Yes.
Do you have a sense of whether, is that cold trap water? Could it be kind of internal water that's trapped in the, I know, some crater peaks have been shown to be sort of relatively water rich, and that's thought to be hydrated mantle material that was uplifted. Do you have a sense of whether that is likely to be cold trap water, or could it be internal water?
Yeah, I definitely did not do that actual study myself, but it's my understanding that this is more likely surface water that was like a cold trap type, based on my understanding of what was done. So no, if there's any kind of uplifted rock water or anything like that, I don't think that would show the same way. So no, but you definitely should ask someone smarter than me. Thank you.
Thanks.
All right, so can we all thank our speakers again?